With more than 6,000 small and microcap companies listed, if you're looking for the next apple at the earliest stage, then Channel Check truly is the orchard. The listed companies support Channel Check, so it's free for you, the potential investor, to gain access to institutional quality research from FINRA licensed analysts, advanced market data, industry reports, news, and a growing catalog of videos and webcasts. Make sure to subscribe to our YouTube channel and register for Channel Check so you're always up to date on what's going on at the small and microcap data place. Welcome to this special edition of the C-Suite series presented by Channel Check and Noble Capital Markets. Noble is an SEC-registered FINRA-licensed broker-dealer and the source of the equity research available on Channel Check. This investor forum at the World Stem Cell Summit presentation features Cellularity. Following a brief overview presentation from founder, chairman, and CEO Dr. Robert Herrera, Noble Biotech Research Representative Greg Oran will moderate a Q&A session. If you registered for this event through channelcheck.com, in the coming days you'll be given an opportunity to get your own questions answered by the company. With that, I am pleased to present Dr. Robert Herrera.
Good morning. I'm Bob Hariri. I'm the chairman, founder, and CEO of Cellularity. Cellularity is a cellular medicine company very much focused on the next evolution of cellular therapy, and I'm very grateful to have the opportunity to speak with you today at the investor forum of the World Stem Cell Summit. As a preface to a broader discussion and questions and answers, I'd like to highlight some important differentiating features of the Cellularity approach to delivering high-performance cell therapies, which clearly define some of the unique attributes of our platform, as well as differentiate our approach to other companies in the industry. These three important things resonate throughout the presentation, so I'd like to first focus on the unique source of cells that Cellularity employs to develop these cellular therapeutic products. Our company is credited with the discovery and defining that the postpartum placenta is a rich source of high-quality stem cells which can be procured and processed under very rigorous controls to deliver unique biological properties, including a high degree of stemness, which I'll speak to in a moment, the unique immunologic characteristics of these cells, which make them the ideal universal donor material for developing off-the-shelf allogeneic therapies, and the breadth with which we can differentiate these cells into products that have a variety of biologically relevant properties. Stemness is a key feature of cells derived from newborns, and stemness can best be thought of as intrinsically advantaged proliferative and synthetic properties, which make them both more potent and capable of mass production and increased durability and performance when delivered clinically. The next characteristic that helps define our company from others in this field is that we have a very, very broad and robust pipeline across a variety of programs and indications. And this provides us a possibility of a variety of value creation events starting as early as the second quarter of this year, carrying through over the next several years, as defined in our pipeline slide here, which shows active programs in a variety of cancers, both hematologic malignancies and solid tumors, as well as degenerative diseases. We've also have activities in infectious disease, and we have a robust program in biological materials as well. And the third characteristic that is, I think, quite defining for our business is that our platform is a true defined and dominant platform. Often companies describe themselves as platform technology businesses when they take a defined asset, either incorporate a specific tool set or a development strategy, and leverage that in one or more indications. In contrast to that, Cellularity is both responsible for the discovery, the definition of the manufacturing and scalability process for turning living cells into broadly deployable medicines. And we have a range of cellular products from immunotherapeutics to pro-regenerative and immunomodulatory products that can be used in a number of indications. These include immune cells such as natural killer cells, T cells, which can be engineered to express a CAR construct, and these are all being deployed in degenerative diseases, infectious disease, and cancer. In the future, Celularity intends to leverage this very, very rich source of cells into a range of engineered and native constructs that have activity in a range of disorders. Celularity, because of this, has the ability to both be an independent, competitive player in the broad area of cellular medicine, as well as to be an ideal partner of choice for companies with products that are approved or in development in these different clinical areas. So please keep these three ideas in top of mind as I speak a little bit further. The superior stemness of cells derived from a newborn, near-term and long-term opportunities in our pipeline, and the ability to leverage the platform into really a very, very wide array of clinical opportunities. So Celularity is currently best described as a clinical stage biotech company, very focused on leading what we see as the next evolution in cellular medicine. And this is very much focused on developing off-the-shelf allogeneic therapies, all derived from this unique source material. The company's origins date back to Anthrogenesis, a company that I founded over 20 years ago, which at the end of 2002 merged into a rapidly growing biotech company, Celgene Corporation, and we built the Cellular Therapeutics division at that time, very much focused on creating a highly proprietary placental cellular platform, and as well as very deep expertise in all of the attributes necessary to turn living cells into medicines. That includes things like process engineering, quality systems, and scalable manufacturing, all of which we believe are central to durable and sustainable success in this business. And then in 2017, after approximately 14 years as part of Celgene Corporation, we were the first and only spin-off company from Celgene prior to its merger into Bristol Myers. The company, Celularity, is currently in the clinic or will be filing very shortly with the FDA to have 4 unique allogeneic placental-derived cell types. These include unmodified natural killer cells, genetically modified natural killer cells, placental CAR T cells, and mesenchymal-like adherent stromal cells derived from the placenta, all of which are being deployed in a range of indications, as I mentioned before, from cancer, including solid tumors, as well as hematologic malignancies, serious viral infections, and a range of degenerative diseases. The company is currently enrolling in 4 clinical trials, and I'll be filing 2 additional INDs this year. We anticipate being in 4 to 5 pivotal or registration-enabling studies by year-end. This clinical pipeline represents a variety of opportunities, what we like to describe as multiple shots on goal, and a clinical strategy that can be readily extended to additional indications we can discuss in greater detail in a few moments. Let me speak in a little more detail about this characteristic of stemness. So it's now increasingly recognized that greater stemness is associated with superior clinical responses to cellular immunotherapy. Celularity's placental cell platform is characterized by this greater stemness, which translates into better proliferative performance both in manufacturing and when delivered as a therapeutic, greater persistence in clinical applications, and these cells, because they're derived from a newborn, an immunologically naive newborn, yield safer, more potent therapies with little or no risk of some of the immunologic adverse effects, including things like graft versus host disease. And we've also shown that we do not elicit a significant host anti-therapeutic or anti-graft response. In fact, in our experience of treating hundreds of patients with placental-derived cells, we have never identified the creation of alloantibodies to these placental products. It's a unique property, and we believe it increases the versatility of using these cells in different applications. So by harnessing the placenta's unique biology, I mentioned to you the stemness. I also mentioned to you the fact that the placenta is nature's universal donor tissue. And by the way, this characteristic of immunologic tolerance inducing effect is obviously very relevant to the development of allogeneic products. And as you can imagine, the placenta is an organ that's produced by the developing fetus, which is the product of the combination of the genetic material of both the mother and the paternal system. So the mother carrying that fetus and its placenta is a 50% donor of that genetic information, and therefore an incomplete tissue match. However, the mother carries that fetus and its placenta for 9 months without rejection. In fact, if you consider that surrogate pregnancy, a mother carries a fetus and its placenta that she's contributed no genetic information to, yet she tolerates it as if it was an endogenous or autologous tissue. That's a unique property that makes for an ideal platform for therapeutics. In addition, the stemness characteristic, which translates into a high degree of proliferative potential, is obviously very relevant to our ability to mass produce these cells, as well as for these cells to have a sustainable effect when administered clinically. If you think about it, cells from newborns, particularly cells from the placenta, give rise to cells which take up residence in the newborn. And those cells have the fidelity and differentiation integrity to be able to be differentiated and expanded over the lifetime of the individual. That proliferative potential translates into a high degree of expansion fidelity in manufacturing and consequently also in the clinic. The recent data from the ASH conference, the American Society of Hematology, has specifically focused attention on this characteristic of stemness. Dr. Stephen Rosenberg has published elegantly that the stemness feature is in fact correlated with a higher degree of clinical performance and better clinical outcomes. And so this platform of placental technology with higher stemness is anticipated to benefit from that particular attribute. So by harnessing this unique biology of the organ, the placenta, and through a very robust procurement and logistical system, which we've developed over the 20 years of our corporate history, we're in a position to be able to develop therapeutic solutions at unparalleled scale, quality, and ultimately economics. Cellularity, in its tenure, both as a private company prior to being part of Celgene, during our time at Celgene Corporation, and since our spinoff in 2017, has developed an extremely powerful, deep and broad intellectual property position, covered by almost 1,000 issued patents, covering everything from composition of matter to methods of manufacturing, analysis, and obviously clinical utility. The other important differentiating factor for Cellularity, which I think in this industry of cellular medicine, which is clearly being recognized as here to stay, deep understanding of how to turn living cells into medicines, basically the expertise around manufacturing, quality systems, is key to being a sustainable, successful enterprise in this field. The key to developing and delivering cellular therapies with quality, consistency, and reliability is really dependent upon that sophisticated manufacturing expertise. The team here at Cellularity has been manufacturing cells under GMP, good manufacturing practices prescribed by the FDA, for almost 15 years. And we've produced multiple cellular products, including immunotherapeutic products, pro-regenerative therapeutic products, as well as biological products from placentas that have been procured in one of the world's most experienced systems ever devised. Cellularity and our predecessor companies have procured cells and other byproducts from over 50,000 newborn donors, and we have in our biorepository here at our headquarters a cryopreservation library of almost 100,000 newborn cell lines derived from this organ. The company had, upon our spinoff from Celgene Corporation, recreated our capabilities by building a purpose-designed and constructed facility for both research development, as well as manufacturing and biorepository operations. Our state-of-the-art facility is designed for parallel commercial production of a variety of cellular therapy products, as well as advanced biomaterials. And we're currently in production at our facility with 3 discrete cell products, natural killer cells, T cells, as well as pluripotent cells, and as well as other biologic products derived from the assembly line of turning living cells into these cellular medicines. So, in summary, the company is poised to be a leader in this industry by advancing the platform of technologies from the placenta, and we are extremely experienced, as can be seen in this slide, with a series of important milestones over the year 2021, and a number of important upcoming milestones, including clinical data readouts and entry into the clinic with some of our advanced programs that we believe position us to be highly competitive in this industry going forward. So in closing, I want to thank you very, very much. And I know we've covered a number of issues, but I'd like to be able to turn this over now to questions. And I'm joined here by our head of investor relations, Carlos Ramirez, who can also be a source of information going forward. Thank you.
Hi, this is Greg Arand at Noble Capital Markets. Thank you, Bob, for joining us today. We appreciate your time, and it was a great, excellent presentation. I do have a couple of follow-up questions regarding your presentation, and this is coming from the audience. A couple of things related to a multitude of indications that you expressed. Can you provide a little more context about the pipeline and when you're releasing more results on the programs?
Certainly. So we're very fortunate at Cellularity because the placental source material provides us with access to the most primitive pluripotent stem cells, which, as you know, can be used to derive a range of mature differentiated phenotypes, including immune cells, and the sheer abundance of the progenitor populations in this organ make it an ideal, highly scalable resource for the mass production of cellular products. And among those products from the placenta, we have advanced to the clinic natural killer cells from the placenta. We are filing an IND to introduce placental T cells, which can be engineered to express a variety of CAR constructs. We have pluripotent placental cells, which are both immunomodulatory and pro-regenerative. And we have a variety of other byproducts of our manufacturing system, which I can get into later. But the key focus and the key thrust of our programs today is in the area of immuno-oncology, where we have completed phase 1 studies using natural killer cells from the placenta that are not engineered. These are native natural killer cells. Our strategy for introducing and going into the clinic first with that product was the natural native function of natural killer cells as a defense system against the transmission of malignancy from the maternal carrier to the developing fetus. So I might spend a couple of seconds speaking about that. One of our early observations after defining that the placenta was a remarkable source of stem and progenitor cells for various purposes was that in addition to this biological stem cell factory that the placenta serves as, it also serves an important role as a defense organ. And what I mean by that is, as you can imagine, human beings devote 9 months of reproductive energy to a single offspring under most instances. Nature is a lot smarter than we are, and evolution has selected for a variety of activities and systems that defend this developing fetus against all types of threats. Those threats may be, for example, infectious disease threats, but they also can be the transmission of cells which might be transformed, thereby putting the fetus at risk. We were fascinated by this many years ago and embarked on understanding the immunobiology of the organ for lots of reasons. To better understand its ability to be tolerated by an allogeneic host, but also because we want to identify what was behind this native ability to defend against transmission of cancer. So just to give you a statistic that's very fascinating, About 1 in every 1,000 pregnant women has some form of cancer during pregnancy. Yet reports of transmission of that maternal cancer to the developing fetus essentially are non-existent. It's so incredibly rare that if a newborn has a cancer that they believe might have been transmitted in utero, it becomes the topic of a case report. We fundamentally believe that the placenta has evolved a part of its immune system, part of its innate immune system that's highly defensive against cancer cells. And when we took that into the laboratory, we identified that placental natural killer cells are an ideal weapon to identify, track to, and destroy cells which express stress antigens, such as cancer cells. And this is a very effective way, we believe, the placenta serves in that defensive role. That's a native property that we leveraged in our first entrant into the clinic in both AML and multiple myeloma. The results of those studies were quite fascinating to us, because in addition to the fact that we saw at the proper dose, at the higher dose, we were getting responses in late stage, unresponsive AML patients. We also recognized that we could maximize and optimize the effect by both serial delivery of these products, as well as fine-tuning the selection of patients for the program. So we're now in a phase 1B study in AML, which we believe will have some clinical readouts in the next several months, which we're very optimistic about, which we believe will show that just the native, unengineered natural killer cells from the placenta are an ideal therapeutic in certain applications. Now, expanding from that, we also recognize, and we have the capabilities and the tools to be able to engineer these natural killer cells in order to enhance their potency, enhance their ability to traffic to specific targets, and enhance their durability. So our next entrant to the clinic, which is a genetically modified natural killer cell, will express a specific construct, a CD16 variant, which will increase the natural killer cell's activity in ADCC anti-tumor effects. We are also working towards advancing a number of candidates in the CAR-NK space. And in parallel to that, because the placenta is such an ideal source of a variety of cells of the immune system, we're advancing technologies in placental CAR T cells as an off-the-shelf, highly scalable, one-size-fits-all CAR T candidate to be an entrant into what we believe will be the next generation or the next iteration of CAR T therapy. As you know, autologous CAR T therapy, as remarkable and as important a breakthrough it has been, certain logistical issues, certain scalability and economic issues call for there to be a next evolution, and we believe placental CAR T cells will be an important part of that. In addition, we've got deep experience using the native immunomodulatory and pro-regenerative activity of the population of pluripotent cells from the placenta to treat certain degenerative diseases and certain autoimmune diseases. And part of the strategy there is an extension of work that was done while we were the cellular therapeutics division of Celgene, where we took placental pluripotent stem cells into patients with moderate to severe Crohn's disease, an autoimmune disease where the immune system attacks the lining of the gut. And as you probably know, based upon observations that women with autoimmune diseases will often go into relatively profound immunologic improvements during pregnancy, which we believe might be related to some of these immunomodulatory cells. And in 3 clinical studies, a phase 1A, a 1B, and a randomized placebo-controlled double-blinded phase 2 study, we saw that simple infusions of these placental pluripotent cells over a period of 5 weeks at the beginning of the trial sustained durable improvements in the disease state as measured by CDAI and other endpoints that were durable out to 1 year. Out to 1 year in patients who no longer were responsive to steroids or biologic agents. Now, we see that as an important indicator that placental cells will have a role in immunomodulation in autoimmune disease, but we also saw it as a basis for taking these same cells into degenerative diseases, some of which are mediated through inflammation, because both as an anti-inflammatory, what we call a cellular immunomodulator, we believe that this platform will be very valuable in those diseases as well. And then you might have also noticed that the company is advancing technologies in the biological materials derived from the placenta during our manufacturing process. We have several products under FDA designation that are marketed, and we also have a developmental program in byproducts of these cells in the form of exosomes. So the company is a very broadly based placental technology company that has the ability to leverage assets in immunotherapy, in degenerative diseases, and the future may hold even brighter opportunities in a much more mainstream set of clinical indications.
Thank you for that. I appreciate the response. I do have a follow-up question on the oncology programs. You mentioned that and you touched on it, but you have one in glioblastoma multiforme, I think, a particularly devastating disease, hard to treat. What's your plans for that and for that program? I think it's in phase 1 or something like that at this point.
It's in phase 1. We're enrolling patients and treating patients as we speak. My background is in neurosurgery, and you're absolutely right, Greg. Glioblastoma is often described as one of the hopeless wastelands of developing novel therapeutics. We became compelled on the basis of preclinical work and on the basis of some very exciting in vitro data on the ability of natural killer cells from the placenta to specifically target glioblastoma cells, that under the right clinical delivery system, we might see significant opportunity to control tumor burden in patients with that disease. Most often, those patients, as you know, are managed after an excisional procedure, which gives us an opportunity to deliver our product both systemically and as you know, at the site of a surgical excision, but also in the case of certain diseases like GBM, blood-brain barrier function allows for the delivery of certain cellular products. But we also are augmenting that with direct intracavitary delivery of the product as well. Now, we're optimistic about this. This, as you know, is a very, very difficult disease to manage. We do believe that failure of endogenous immunity plays an important role in glioblastoma, both metastasizing as well as continuing to develop. And so we are optimistic that at the right dose, with the right mode of delivery, we will get enough anti-tumor effect at the site of these malignancies to get better control than what's afforded by other agents.
One thing you touched on also in a slide was the brand new research and manufacturing facility. What was the incentive or need to take on this big investment so at this point in time in your life cycle?
Greg, that's a really important question. I'm very glad that you asked it. So if you go back in our company's history, and although we are currently, as we're moving towards being a public company from this short tenure as a private company after our spin-off from Celgene, our company has a 2-decade-long history of not only advancing the discovery research that led to selection of the placenta as the source material, but also we recognize that as important and as powerful as the cellular platform is, the process of manufacturing and turning these cells into highly scalable cellular products is also part of the product as well. At Celgene, as the Cellular Therapeutics division, we recognize that turning over one of the most critical parts of developing an actual product, a cellular product, to a third party without the opportunity to do the innovative work, to do the discovery work and the development work, actually would shortchange our control of that technology. And we recognize that in the methods of isolating, characterizing, culture expanding, and then selecting cells for therapeutics, there is a rich opportunity to enhance the intellectual property portfolio, but also to fine-tune the methods of production in order to maximize the clinical attributes of the product. As an example, it's not that difficult to take a cell, hand it off to a third party, and have them culture expand it. But to do so with careful attention to selecting for the phenotype and the characteristics of the phenotype of the finished product that will maximize the predictable performance of the cell in the clinic, to me, was essential. As an engineer by training, I may be a neurosurgeon and a scientist, but as an engineer by training, it was discomforting not to control what I believe was the single most important part of producing a product. So at Celgene, we built all of the cellular manufacturing infrastructure, which gave rise, for example, to Celgene's entry into autologous CAR T therapy. And in fact, much of the infrastructure which we developed now resides at Bristol Myers, the company that merged with Celgene upon our spinoff. We believe that it was central to our success because we're so comfortable that our platform is the best platform in the industry, the placental platform. We were so convinced that deep expertise and deep proprietary control of manufacturing would help us play a leading role in this field. So the benefit of having done this before, having built manufacturing capabilities, having built teams in quality systems, in analytics, et cetera, having done that, we were in a perfect position to design and build our new headquarters, which houses 15 independent GMP manufacturing suites. We did so with specific attention to making sure that we always meet or exceed the highest standards of the regulatory community, that we meet or exceed the highest failure mode analysis. So, for example, as you know, many companies in this field of cellular medicine have suffered near fatal failures of manufacturing, in some cases setting companies back a year or more. We never wanted that to be a problem. So this facility was designed to allow us to parallel manufacture to commercial scale multiple products under multiple shifts and allow us to control not just the quality of the end product, but every innovation and invention that goes into getting there. So it was a big investment, Greg. You know, you hit the nail on the head. One might say it's an important calculated risk, but we're designed for success here. This company is not a one-trick pony that is looking to take a single program through to registration and then cut a deal to take that into commercialization. Our objective is to build a sustainable, industry-leading company grounded in our technology that has the ability to always innovate so that we can drive up quality, drive up clinical performance, drive down cost, and drive up quality. And right now, fortunately, in this environment, we're ideally poised to do that.
Terrific. You reduce the risk in manufacturing by taking it in-house. That's a very laudable position. Where do you see Cellularity in the next couple of years, assuming you receive approval on one of your cancer registrations? And also related to that, what other types of programs or indications and diseases are you trying to attack? You have a pretty full pipeline right at the present, but do you have other areas you are seeking to work into?
So, Greg, I'm convinced. I've been doing this for almost 25 years. I'm convinced we have gone past that important point where cellular medicine is now established as a tool that will be used not just in serious life-threatening diseases like cancer, but will eventually migrate into the more common but clearly troublesome unmet medical needs, from cardiovascular disease to metabolic disease to even things like improving and maintaining human performance. Our strategy, because we have such a broad platform, was to go after the highest unmet medical needs in diseases where, for historic purposes and so on, we believe there was a highest probability of regulatory attention to get a product across the regulatory finish line as a way of catalyzing our entry back into the clinic in more mainstream diseases. You know, the Crohn's program, I mean, one could argue that our data in Crohn's is compelling enough to focus all of our attention there. But we don't see Crohn's or autoimmune disease or degenerative disease as an end game for us. We see all of these things as simply building on the fundamental power of this placental technology, building on the power of our 20 years experience in the field, and ultimately we want to provide entrance into a variety of disease indications. As you probably remember, we were the first company to have an IND for a cellular therapeutic product in COVID. Now COVID has taken a lot of twists and turns, and obviously with the advent of vaccines and the deeply competitive nature in that field, you know, we recognize that our work in COVID could be an entry to transform how infectious diseases are treated. And I'll be very clear with you. I believe that the treatment of viral illnesses and serious infectious diseases deserve a revolution. And I believe that revolution is going to be immunotherapy. Immunotherapy is intuitively logical way to treat infectious diseases, and the same stress antigens recognized by our NK cells in cancer also recognize express stress antigens on virally infected cells. So the company is taking into account our ability to leverage success in cancer, a registration in cancer, to move aggressively into these other areas. And again, the company is built to be a highly sustainable business, and we're very optimistic that a registration will open the floodgates for other clinical opportunities.
You have a tremendous opportunity. I appreciate that answer. Importantly, for a lot of people looking at Celularity, I believe you are in what's called the de-SPACing process. When do you expect this to occur? When you get your ticker symbol, the timing, how does this all work out?
So, as many of the audience probably recognize, SPACs are an alternative pathway to public that really became extremely popular over the last 2 years. We were very fortunate. We were moving towards a traditional crossover to IPO round, and a very, very important special purpose acquisition corporation became known to us, and we saw a remarkable fit, and so we chose that pathway. During the process of moving towards closing or de-SPACing, the Securities and Exchange Commission enacted certain new regulatory requirements for accounting of these special purpose acquisition corporations, which we have gone through, and I believe we've met all of the requirements. So we're anticipating being able to de-SPAC in the next several weeks. I would estimate that by no later than the early part of July, we should be through the de-SPAC process, trading as a public company under the symbol CELU, which actually, for me, it's gratifying. I spent the vast majority of my career in industry at Celgene, CELG, and you probably know that Celgene was a spinoff of Celanese. So, the Celularity misspelling is very intentional, Greg. It's to show the continuity between Celanese to Celgene to Celularity. So we're very, very grateful that we got that symbol. Today, trading in the GX SPAC, which is under the symbol GXGX, is ongoing. And, I'm extremely optimistic as a newly minted public company, we're going to be able to put that capital to good use and aggressively prosecute our clinical programs.
Excellent. So you said the next several weeks. So you're going to become public sometime in July. Is that correct then?
Yeah, I believe in the early part of July, we'll get through this de-SPAC and be a newly minted public company.
Excellent. One other question I think that was touched on, you mentioned human performance and longevity. What do you mean by that? exactly.
So you're touching on you can tell by all the gray in my beard, Greg, that I'm worried about maintaining performance. I'm a surgeon by training. Earlier in my life, I was an athlete. Everything hurts now. I'm in my 60s. Everything hurts. I can tell you that we did some very exciting work many years ago, and we're not alone. That has defined a relationship between the quality of your stem cell reservoir that resides in virtually every organ and tissue of your body, and your ability to maintain high performance renovation and renewal of your tissues during your lifetime. And it's associated with better performance. It shouldn't be a surprise. If you've got good connective tissue, soft tissue, muscular tissue, those systems operate at high efficiency, high integrity. I'm convinced that the future in cellular medicine, once we get through applying specific discrete cell types in things like immunotherapy, will be to recharge the regenerative engine that helps maintain performance as we age. I don't like the terms longevity, and I'll tell you why. When I give a talk about this and I say, how many in my audience here want to live to 150? The simple concept of adding years to life isn't very attractive because the mindset is a 150-year-old individual is extremely frail and feeble and so on. But when I ask folks, how many of you would like to live another 75 years exactly the way you are right now, everyone raises their hand. My belief is that the way to do that is to maintain the quality and integrity of this stem cell reservoir, which is your regenerative engine. It's what keeps you humming and ticking. And as a lifelong aviator, you know how I fly airplanes that are 40 or 50 years old? They get rebuilt every 10 to 12 years, and they get rebuilt back to perfection. I believe the future in cellular medicine may include a strategy like that.
Excellent response. That sounds very interesting. It opens up the door for all kinds of opportunities there as well, too, to extend life. Especially when it comes to brain function. I think that's somewhat of a concern for a lot of people, not just the physical aspects, but also the mental aspects. I appreciate that indeed.
Greg, you're so right, because the truth is, high performance, healthy aging, you've got to maintain high performance mobility, high performance cognition, youthful aesthetics, and a high performing immune system. If you can do that, extra years are worth it.
Thank you for joining us for this C-Suite interview presentation brought to you by Channel Check. Visit our YouTube channel for more interview content, as well as virtual roadshows and conference presentation replays. New content is added regularly, so subscribe below to stay up to date. Visit channelcheck.com or click the link in the description to access equity research, news, and advanced market data on this and the 6,000 other small and microcap companies listed.